When you finally get a chance to sleep in, like you log 8, maybe 9 straight hours.
Oh, yeah. The kind of sleep where you wake up and you should theoretically feel like $1,000,000.
Right. Exactly. But instead, you swing your legs out of bed and you are just, you're just heavy.
Yeah.
You reach for a thought, like trying to remember what you need to do today, and you just find fog. And by, 2 in the afternoon, your body is essentially screaming at you to lie down, even though you've spent the entire day just sitting at a desk.
It is a totally maddening experience for a lot of people, especially because, well, the first thing you do is go to your doctor.
Right, you want answers.
Exactly. And they run the standard blood panels. They check your thyroid, your iron levels, vitamin B. They do a complete metabolic panel. And then a few days later, they call you back and say, great news. Everything is perfectly normal.
But you don't feel normal. You feel like your battery is 2% and the charger is completely broken.
Yeah.
And that disconnect between the standard blood work looking fine and a patient feeling just totally drained, that's exactly what we are exploring today. We are going off the standard map for this deep dive.
We really have to.
Yeah, instead of looking at the macro systems like the organs or the blood, we are going deep underground to look at your cellular power grid. We're talking about the mitochondria.
It's a completely necessary paradigm shift because for decades, the medical focus for fatigue has been very top-down. Like, if you are tired, let's look at the heart pumping the blood or the lungs getting the oxygen.
Makes sense on the surface.
Right. But the reality is, if we want to understand why our internal batteries break down and, more importantly, how to actually fix them, we have to go down to the microscopic level because what happens at the micro level dictates literally everything you experience at the macro level.
I was reading through the notes and the protocols you shared with me,
Yes.
Stuff from integrative clinics like RTHM and SI Medical, plus like a mountain of clinical trial data on post-viral fatigue.
Yeah, there's so much data now.
There is. And the analogy that kept coming to my mind was a major city. When we think about the health of a city, we look at the traffic, which is our blood flow.
Right.
And we look at the skyscrapers and the infrastructure, which are our muscles and bones. But none of that matters if the power plants go offline. You can have the best roads in the world, but if the grid goes dark, the whole city shuts down.
That analogy holds up incredibly well, actually.
Oh.
But I want to elevate the stakes even higher than just power generation.
Okay, higher how?
Well, if you took high school biology, you were probably taught that classic phrase, right? The mitochondria is the powerhouse of the cell.
Right, it's literally the one thing everyone actually remembers from 10th grade science.
Exactly. And it is a massive undersell. Calling them just powerhouses ignores their true role.
Yeah.
They are the ancient, centralized decision-making hubs of human biology.
Decision-making hubs.
Yeah. They don't just supply energy. They dictate how your immune system responds to threats. They manage calcium levels in your cells, which controls muscle contraction. They even produce their own local hormones.
Wow, I had no idea they did all that.
Oh, it gets wilder. Because most profoundly, they control a process called apoptosis, which is programmed cell death. The mitochondria are the entities that ultimately decide if a cell gets to keep living or if it must destroy itself for the greater good of the body.
Wait, they can just pull the plug on the whole cell?
They absolutely can. If they detect too much damage, they release a protein called cytochrome C, which essentially triggers a self-destruct sequence. So they aren't just mindless engines burning coal,
Yeah.
They are the intelligence running the grid.
Okay, that changes the perspective entirely. But to really understand how these microscopic power plants got so much authority over our bodies, we need to go back. Because I was reading the evolutionary background in those research reviews, and frankly, it sounds like science fiction.
It really does.
Where did these organelles actually come from?
Well, we have to go back billions of years to a period before complex multicellular life even existed. The leading, widely accepted scientific framework for this is called endosymbiotic theory. Imagine an ancient single-celled organism, a prokaryote. It's floating around in a primordial soup, and it essentially swallows another smaller bacterium.
Like a predator eating prey.
Exactly like that. But here's the cosmic accident. For whatever reason, the host cell didn't digest it. The smaller bacterium was unique. It was aerobic.
Meaning it used oxygen.
Right, meaning it could use molecular oxygen to generate massive amounts of energy, and it was even producing early forms of melatonin. So instead of destroying it, the two cells struck a biological bargain, a symbiosis.
Oh, I see.
Yeah, the large heart cell provided a safe, nutrient-rich environment, and in exchange, the smaller bacterium paid rent by churning out energy.
Are you saying that right now I am walking around with ancient bacteria living inside my cells?
I am, and we know this for an absolute fact because mitochondria still possess their own unique DNA.
Hold on. Their own DNA.
Hmm.
Separate from the DNA in the nucleus that makes me, well, me.
Completely separate. Your nuclear DNA, the human genome that dictates your eye color and height, is a mix from both your parents. But mitochondrial DNA is totally distinct.
How so?
It's circular, much like bacterial DNA, and it is maternally inherited. You receive it strictly from your mother, passed down through the egg cell in an unbroken chain going back hundreds of thousands of years.
That is just wild. So we are basically highly evolved transport vehicles for this ancient bacteria.
Pretty much.
What is the scale of this operation inside a single cell? Are we talking about one or two of these power plants per cell?
Oh, not even close. The number scales based on the energy demand of the tissue. So a skin cell might have a few hundred. But in highly metabolically active tissues, like your skeletal muscles, your heart, your liver, and your brain, a single cell can contain between 1,000 and 2,500 mitochondria.
Thousands per cell.
Yes. In heart muscle cells, mitochondria can physically occupy up to 25% of the entire volume of the cell.
A quarter of the heart cell is just engines.
Yes. To put the sheer industrial scale into perspective here, under resting conditions, just sitting on the couch breathing, the human body processes roughly 1 kilogram of molecular oxygen every single day.
Wow.
Yeah, strictly to fuel these organelles.
1 kilogram of oxygen gas. That sounds like a heavy industrial process. So let's look inside the factory. If I am breathing in that oxygen and I am eating, say, a piece of toast for fuel, how does the mitochondrion actually convert that into biological electricity?
So we have to zoom in on a piece of biological machinery called the electron transport chain, or the ETC.
Okay.
If you look at a mitochondrion under an electron microscope, you'll see it has a smooth outer membrane, but inside it has an inner membrane that is folded back and forth on itself. Those folds are called cristae.
Cristae. Got it.
And embedded right in the wall of that inner membrane are 4 massive protein structures, unimaginatively named complex 4, 2, 3, and 4.
Okay, so a folded wall with 4 stations built into it.
Right. When you digest that toast, your body breaks it down and extracts high-energy electrons. These electrons are shuttled to the mitochondria and handed off to complex P and complex 2.
Like delivering coal to the plant.
Exactly. Now think of the ETC like a bucket brigade, or maybe a series of microscopic specialized toll booths. The electrons are passed down the chain from complex 1 to 3 to 4, and as they are handed down, they release a little bit of energy at each step.
What is the cell doing with that energy? Is that the electricity we use?
Not yet. The complexes use that energy to physically pump positively charged protons from the inside of the mitochondrion out into the space between the inner and outer membranes.
Wait, so they're moving electrons down the line just to power a pump? It sounds like they're creating a pressure buildup.
Yes.
Like pumping water up a hill to fill a reservoir behind a dam.
That is the exact mechanism. They are creating what we call an electrochemical proton gradient. You have a massive buildup of positive charge on one side of the membrane and a negative charge on the other. The protons desperately want to rush back across to equalize that pressure.
So how does the cell harvest that pressure?
Through a 5th station called complex V, or ATP synthase. And this is honestly one of the most beautiful structures in biology. It is literally a microscopic physical turbine.
A turbine?
Yeah, it has a rotor and a stator. The cell opens the gate at ATP synthase, and all those trapped protons rush through it, just like water rushing through a hydroelectric dam.
Oh, wow.
The physical force of the protons makes the turbine spin at thousands of revolutions per minute.
Wait, it physically spins inside the cell?
It physically spins, and the mechanical energy of that spinning turbine forces an inorganic phosphate molecule onto a molecule called ADT, turning it into ATP.
Adenosine triphosphate.
Exactly. ATP is the universal energy currency of all life on Earth. Every time your heart beats, every time a neuron fires, to let you understand my words right now, it is fueled by ATP generated from those spinning turbines.
I am having one of those moments where biology sounds completely made up. I mean, I have billions of microscopic hydroelectric dams spinning inside me right now.
It's incredible to think about.
It really is. But here's the thing about industrial power plants. Whether they are coal or nuclear, they are never 100% efficient.
Right.
There's always pollution, always a thermodynamic tax. If we are moving all these high-energy electrons around at this massive scale, what is the biological cost of doing business?
You've identified the central thermodynamic paradox of human life. The system is incredibly efficient, but it is not perfectly clean. As those electrons are being tossed down the bucket brigade, occasionally one fumbles.
It just drops it.
Yeah, it slips out of complex I, or complex 3. And when a free electron escapes, it reacts directly with the oxygen floating around in the cell. This creates what we call reactive oxygen species, or ROS.
ROS. Give me an example. What does it actually mean?
The most common ones are superoxide anions and hydrogen peroxide. They are highly volatile molecules that have an unpaired electron. Because they're unstable, they will violently rip electrons off of anything nearby just to stabilize themselves. We refer to this entire process as oxidative stress.
Okay, this brings me back to the power plant analogy, specifically like a nuclear reactor. The reactor generates massive, totally vital power for the city. But if the core isn't properly contained, it leaks toxic radiation into the immediate surrounding area.
Yes. In this case, the ROS acts exactly like radiation. It damages the lipids in the mitochondrial membrane, it degrades the proteins in the electron transport chain, and critically, it damages the mitochondrial DNA itself.
Oh, no.
Yeah, and because that DNA is sitting right inside the matrix, mere nanometers away from the electron leak, it takes heavy collateral damage over the years.
But surely the body has a defense system, right? A nuclear plant has cooling rods to absorb the excess heat and radiation.
We do. We have an endogenous, localized antioxidant defense system. The cell produces its own cooling rods. Things like glutathione, coenzyme Q10, and an enzyme called manganese superoxide dismutase.
That's a mouthful.
It is. But their sole job is to jump on those free radicals and neutralize them before they damage the machinery.
So what happens when those cooling rods degrade? Like, let's say I'm getting older, or my diet is terrible, and my antioxidant defenses weaken.
Then you enter a vicious pathological feedback loop. Without enough cooling rods, the ROS radiation damages the mitochondrial DNA.
Right, the blueprints.
Exactly. The damaged DNA then produces faulty proteins for the electron transport chain. Now you have a leaky, inefficient turbine. Because it's inefficient, it drops even more electrons, creating even more ROS, which causes even more damage.
So it's a death spiral for the cell.
Exactly. When we look at the biology of aging, a massive portion of it is simply mitochondrial decay. In fact, if you look across all mammalian species, the robustness of a species' antioxidant systems and their ability to contain ROS leakage is one of the strongest biological predictors of their maximum lifespan.
That is fascinating.
Aging is fundamentally the slow, decades-long accumulation of this structural and genetic damage to the power grid.
Okay, I think I have a solid grasp on how the grid works and how it naturally rusts and decays over a lifetime, but that's a slow process. What I really want to dig into are the clinical case studies you shared regarding complex chronic illnesses.
Let's do.
We are talking about long COVID, MECFS, which is myalgic encephalomyelitis, often called chronic fatigue syndrome, and things like dysautonomia. In these patients, the grid doesn't just rust over 80 years. It seems to catastrophically crash overnight. What is happening there?
This is a critical distinction. In aging, we see wear and tear. In long COVID and MECFS, we are looking at a profound acquired energy crisis. The grid has been hijacked and physically dismantled by a pathogen.
Let's talk about the SARS-CoV-2 virus, because the data on this is just staggering. The virus isn't just irritating the lungs, it's actively targeting the power supply.
Yes, viruses are intracellular parasites. They cannot replicate on their own. They need massive amounts of energy to mass produce their viral machinery. So SARS-CoV-2 literally enters the cell, locates the mitochondria, and hijacks the machinery for its own replication.
Wow.
And the physical damage it leaves behind is visible under an electron microscope.
What does a hijacked mitochondrion look like?
Well, in biopsies from long COVID patients, researchers don't see healthy, bean-shaped organelles with tight internal folds. They see swollen, fragmented mitochondria. Those beautiful internal cristae, the folds where the electron transport chain lives, are completely disrupted or obliterated.
So using our dam analogy, the pathogen has literally blown up the concrete walls holding back the water.
Precisely. The structural scaffolding is ruined. Without those tight folds, the complexes of the electron transport chain fall out of alignment. They can't hand off the electrons efficiently.
Because they're physically too far apart now.
Exactly. The proton gradient collapses. The result is a massive, immediate drop in the cell's ability to produce ATP.
Okay, I want to connect this to what a patient actually feels, because earlier we talked about that heavy fog brain feeling. But in MECFS and long COVID, There is a specific hallmark symptom called post-exertional malaise, or PE.
Yes, PM.
I hear this term a lot, but I want to understand the mechanics of it. What exactly is PE on a cellular level?
PM is a catastrophic bioenergetic bottleneck. Let's look at a healthy person first. If you decide to jog up a flight of stairs or even just focus intensely on a complex spreadsheet, your brain and muscles demand more energy.
Right.
In a healthy cell, the mitochondria sends a demand and instantly scale up ATP production. The turbines spin faster.
Right. The power plant fires up a reserve generator to handle the spike in demand from the city.
Exactly. But in a patient with ME/CFS or long COVID, the mitochondria are structurally damaged. They have no reserve capacity. When the patient tries to walk up the stairs, the cell demands ATP, but the damaged power plant simply cannot scale up. It hits a hard, unyielding ceiling almost immediately.
But the cell still needs energy just to stay alive.
Yeah.
If it doesn't get it, doesn't it die? What does the cell do if the main power plant fails?
It panics. It realizes the mitochondrial aerobic engine is failing, so it pulls the emergency cord and shifts to a backup system called glycolysis.
Glycolysis, let's break that down. I've heard the term, but I don't really know the mechanism.
Glycolysis is a much older, primitive way of making energy that happens in the cellular fluid outside the mitochondria. It doesn't require oxygen. It rapidly ferments glucose to make ATP.
That sounds like a good backup plan. Why is it a problem?
Two reasons. First, it is incredibly inefficient. A healthy mitochondrion can extract roughly 36 molecules of ATP from 1 molecule of glucose. Glycolysis only extracts 2.
Oh, wow. So you blow through your cellular fuel reserves almost instantly, just trying to maintain baseline function.
Exactly. And second, glycolysis produces a massive amount of metabolic waste, specifically lactate and hydrogen ions, which flood the cell.
So they are running on a dirty, inefficient backup generator that basically fills the building with toxic smoke.
That is a perfect way to describe it. The rapid depletion of fuel and the toxic buildup of intracellular acid paralyze the cell. This is the crash. This is post-exertional malaise.
Wow.
Yeah, it can take days, sometimes weeks, for the cells to clear that lactate and slowly rebuild their ATP pools.
I am so glad we are detailing this because there is such a stigma around these invisible illnesses. A patient says they are tired, and a well-meaning doctor or family member says, oh, you're just deconditioned. You just need to push through it. Go for a walk. Do some graded exercise.
And that advice is biologically dangerous. This is not deconditioning. Deconditioning is a slow loss of muscle mass. PEM is a molecular failure of energy metabolism.
Right. It's totally different.
When you tell a patient with damaged mitochondria to push through it, you are forcing their cells deeper into glycolysis, generating more acid, and creating massive amounts of ROS that further shred whatever healthy mitochondria they have left. You are actively injuring them.
It's a horrifying cycle.
Yeah.
But the data shows it doesn't just stop at low energy. There's a systemic inflammation loop that gets triggered. How does a lack of energy cause inflammation? Is it the ROS leaking out?
It's worse than just ROS. Remember the evolutionary history we discussed earlier?
That mitochondria are essentially ancient bacteria.
Right. Well, when these swollen, virus-damaged mitochondria start to break apart, they literally rupture. And when they rupture, they spill their inner contents, including fragments of that circular bacterial-like mitochondrial DNA, out into the cellular fluid and eventually into the bloodstream. We call this cell-free mitochondrial DNA.
Oh, wow. I see where this is going. If my immune system is scanning the blood.
Your immune system is constantly scanning for pathogens. White blood cells have receptors specifically designed to detect bacterial DNA. When your damaged mitochondria spill their DNA into the blood, the immune system detects it and sounds the alarm. It thinks we have a massive systemic bacterial infection happening right now.
So the body starts attacking itself.
It triggers a very specific defensive complex called the NLRP3 inflammasome.
The NLRP3 inflammasome. That sounds intense. What does it actually do?
Think of an inflammasome as a biological siren. Once activated, it floods the body with highly aggressive inflammatory cytokines. These are signaling molecules that tell the immune system to go to war. This causes systemic inflammation, microvascular clotting, and tissue damage.
Which in turn damages more mitochondria, which leak more DNA, which triggers more inflammation. It's a self-perpetuating loop.
Yes.
No wonder people are sick for years. The immune system is constantly responding to a phantom bacterial infection that is actually just their own broken cellular engines.
Precisely. It is a unifying mechanism in many chronic illnesses, driving the neuroinflammation, the joint pain, and the chronic immune activation that these patients suffer from daily.
Okay, if the immune system is in a constant state of panic and the skeletal muscles are constantly crashing into glycolysis because their engines are broken, what is happening to the brain?
Yeah.
Because the brain requires a massive amount of power, right?
The brain is arguably the most bioenergetically demanding organ in the body. It weighs roughly 2% of your total body mass, but it consumes 20% of your daily energy. It is incredibly vulnerable to any disruption in the grid.
I was looking at a clinical trial you included in the sources, one that focused on older adults experiencing cognitive decline, and it utilized a supplement called GlyNAC. Before we get to the results, break down what GlyNAC is.
GlyNAC is a combination of two amino acids, glycine and N-acetylcysteine. The reason it's so important goes back to our nuclear cooling rods.
Right, the antioxidants.
Exactly. We mentioned glutathione earlier as the master intracellular antioxidant. As we age or deal with chronic inflammation, our glutathione levels absolutely plummet, leaving the mitochondria defenseless against ROS.
So why not just take a glutathione pill? Why take GlyNACs?
Because oral glutathione breaks down in the digestive tract very poorly. But if you give the cell the raw precursor building blocks, which are glycine and N-acetylcysteine, the cell will rapidly absorb them and synthesize fresh glutathione directly inside the cytoplasm where it's needed most.
Okay, so researchers gave GlyNAC to these older adults. What were they looking for?
They were using advanced tracer methodologies to observe glucose metabolism. They literally tracked where the sugar was going in the body. And what they discovered is a phenomenon they termed the brain glucose steal.
The brain glucose steal. That sounds like a bio-thriller movie title. Who is the thief and who is the victim?
The victims are the neurons in the brain. The thieves are the peripheral organs, specifically the skeletal muscles and the liver.
How does that work?
Here is the mechanism. As people age, the mitochondria in their muscles become highly impaired due to oxidative stress and glutathione deficiency. Because the muscle engines are inefficient, they become bioenergetically desperate. To avoid dying, the peripheral organs start aggressively upregulating their glucose transporters, consuming and hoarding the glucose in the blood that was originally slated to travel up to the brain.
Oh, this brings the city analogy perfectly to life. It is exactly like rolling blackouts.
Right.
The outer suburbs, the muscles, are so desperate for power, so inefficient, that they are siphoning electricity off the main grid just to keep the streetlights on.
Right.
And they are stealing that power directly from the downtown hospital, which is the brain.
That is exactly what is happening. The brain requires a steady stream of glucose to maintain cognitive function. When the muscles steal it, the brain experiences a localized energy crisis. The result: brain fog, memory issues, slower processing speeds, and cognitive decline.
So how did the GlyNAC intervention affect this? Did restoring the cooling rods stop the robbery?
It did. The trial supplemented the older adults with GlyNAC for 24 weeks. This successfully ramped up intracellular glutathione, which neutralized the ROS. Once the oxidative stress was lifted, the existing mitochondria in the liver and muscles could finally operate efficiently again.
And because they were efficient, they didn't need to hoard the sugar.
Exactly. The glucose steal stopped. The glucose became available in the bloodstream, traveled to the brain, and the neurons were finally fueled properly.
That's amazing.
The clinical outcomes were remarkable. The older adults showed significant, measurable improvements in cognition, memory, and executive function. But they also saw physical improvements like increased gait speed and grip strength because the muscles were functioning properly too.
That is such an elegant solution. I mean, you aren't giving them a brain stimulant. You were just fixing the efficiency of their leg muscles so the brain can eat.
Right. It's fixing the root cause.
But as I read the study, there was a major catch at the end of the trial, wasn't there?
Yes. They included a 12-week washout period where they stopped the GlyNAC supplementation.
And let me guess, the grid collapsed again.
Unfortunately, yes. Without the precursors, glutathione levels dropped back to baseline, the oxidative stress returned, the peripheral mitochondria lost efficiency, the glucose steal resumed, and the cognitive defects came right back.
Wow.
It highlights that maintaining metabolic health isn't a one-and-done cure, you know? It is a continuous daily requirement to manage the thermodynamic tax of living.
Okay, we've identified the problem. We have heavily damaged, inefficient, ROS-leaking mitochondria, draining our resources, causing systemic inflammation, and stealing fuel from the brain. If I want to fix this, I can't just try to build new power plants right away. I need to deal with the rubble first. How does the body tear down a condemned building?
The body has an incredibly elegant, highly selective cellular recycling system called mitophagy.
Mitophagy?
Yes, mitophagy is the specific process of identifying broken mitochondria, dismantling them, and clearing away the debris so it doesn't cause inflammation.
And one of the most exciting tools for this, according to the research you provided, is a compound called Urolithin A, or UAKKA. But reading the origin story of Urolithin A, it's just bizarre. You can't just eat a food that has it.
No, you can't. Urolithin A is what we call a postbiotic metabolite. It does not exist natively in any food. To get it naturally, you have to eat foods rich in complex polyphenols, known as allagitannins and ellagic acid. We find these in pomegranates, walnuts, strawberries, and raspberries.
Okay, so I eat a bowl of pomegranate seeds. What happens next?
Those polyphenols travel down to your gut. There, very specific strains of bacteria in your microbiome have to ferment them and synthesize them into urolithin A.
Wait, I have to rely on my gut bacteria to make the medicine for my mitochondria.
Yes.
What if I don't have those specific bacteria?
Then you don't get the urolithin A. It is a severe biological bottleneck. Clinical research indicates that only about 30 to 40% of the global population actually possesses the right microbiome composition to convert these polyphenols into therapeutic levels of urolithin A.
Only 30 to 40%.
Yeah. And if you are dealing with a chronic illness or if you've been on multiple rounds of antibiotics, your gut dysbiosis makes that conversion rate effectively zero.
That is incredibly frustrating. The body has a cleanup mechanism, but the activation key is locked behind a gut bacteria that most people don't even have.
It is frustrating, but there are workarounds now.
Right. So let's assume I do have the bacteria. Or, more practically, I take a direct, purified urolithin I supplement to bypass the gut entirely. Once UA is in my bloodstream, how does it actually trigger the demolition of these broken mitochondria?
It acts as a pharmacological trigger for a very specific molecular cleanup sequence called the PNK1 Parkin signaling pathway.
Okay, PINK1 and Parkin. Let's break those down. I am a broken, failing mitochondrion that is leaking ROS. What happens to me?
When you start failing, you lose your internal membrane potential. Basically, your electrical charge drops. The cell constantly monitors this. When the voltage drops, a specialized protein called PNK1 accumulates heavily on your outer surface.
It's like painting a big, bright red X on the door of a condemned building.
That's a perfect visualization. Once that red X is painted, it acts as a distress signal that recruits an enzyme from the cellular fluid called Parkin.
Parkin.
Right. Parkin arrives at the damaged mitochondrion and does something critical. It slaps a ubiquitin tag on the outer membrane.
A ubiquitin tag? What is ubiquitin?
Ubiquitin is a small regulatory protein. When a molecule is tagged with ubiquitin, it is the cellular equivalent of the kiss of death. It is a literal homing beacon that calls in the cellular garbage truck, which is a massive structure called the autophagosome.
So PNK1 marks it, Parkin tags it, and the autophagosome destroys it.
Yes. The autophagosome swoops in, entirely engulfs the broken mitochondrion, and uses powerful acids and enzymes to digest it down to its basic amino acids and lipids. It completely removes the source of the ROS leakage and recycles the raw materials. Urolithin A is one of the most potent known activators of this entire PNK1 Parkin process.
I am looking at the clinical data on this, and it is honestly hard to believe. In 2022, there was a randomized, double-blind, placebo-controlled trial published in JAMA Network Open.
Oh, the Urolithin A study.
Yeah. They took older adults and gave them 500 to 1,000 milligrams of direct urolithin A daily. And the results? They saw a 12% improvement in hamstring leg muscle strength and a clinically meaningful increase in their VO2 peak, which measures aerobic endurance.
Yes, and I want to emphasize the most crucial variable of that trial. They achieved those gains in muscle strength and cardiovascular endurance without any change in their exercise habits.
Let me stop you. I am deeply skeptical of magic pills that build muscle without exercise. Are you saying they just sat on the couch, took a pill, and their hamstrings got stronger? How is that physiologically possible without tearing muscle fibers in a gym?
It isn't that they grew massive new muscle fibers like a bodybuilder. It's about the quality and efficiency of the existing tissue.
Okay.
Imagine a factory where half the machines are broken and smoking. The factory's output is low. Urolithin A came in and cleared out all the broken machines. The remaining healthy machines could finally operate at 100% capacity without being choked by toxic smoke and inflammation.
Oh, no.
By simply removing the metabolic drag of the damaged mitochondria, the baseline strength and efficiency of the muscle skyrocketed.
Okay, that makes sense. It's not magic, it's just ruthless efficiency. And the notes mentioned it also helped with joints.
Yes, in osteoarthritis models. Because urolithin A clears out the ROS leaping mitochondria, it halts the localized inflammation in the joint capsule. This prevents the degradation of the extracellular matrix and preserves the cartilage. It is a masterclass in why removing cellular trash is step one in any metabolic rehabilitation protocol.
Okay, so the demolition crew has come through. The PNK1 Parkin pathway has cleared out all the condemned buildings and the smoke is cleared. But now we have empty lots. How do we restock the city with brand new, highly efficient power plants? We can't just run on half a grid.
Exactly. Once mitophagy is complete, you must trigger the opposing process, mitochondrial biogenesis.
Biogenesis.
Yes. This is the literal transcription of new mitochondrial DNA and the physical construction of new organelles. And the master architect of this entire building project is a transcription coactivator called PGC1 alpha.
PGC1 alpha. The architect. How do I get the architect to start drawing up blueprints? Because the supplement sources heavily pushed NAD plus precursors like NMN and NR, along with something called resveratrol. How do these pieces fit together to start construction?
They work in a highly synergistic loop. PGC1 alpha is the architect, but it usually sits inactive in the cellular fluid. To wake it up, you need a formin. In the cell, that formin is an enzyme called SIRT1.
SIRT1.
SIRT1 is widely considered a master regulator of metabolic health and longevity.
Okay, so SIRT1 tells PGC1 alpha to get to work. How do we wake up SIRT1?
SIRT1 is entirely dependent on a molecule called NAD+, RTS, and NAD+ is the fuel that allows SIRT1 to function. The problem is, as we age or deal with chronic stress, an enzyme called CD38 aggressively destroys our intracellular NAD+ levels. The fuel tank basically goes to zero.
So by supplementing with NAD+ precursors like nicotinamide mononucleotide, NMN, or nicotinamide riboside, NR, we're essentially restocking the cellular fuel tank to allow SIRT1 to operate.
Precisely.
Okay, so NMN fuels the formin. Where does the resveratrol come in? I know it comes from grape skins and red wine, but what does it do molecularly?
Resveratrol acts as an allosteric activator. It is a signaling molecule that binds to SIRT1 and alters its shape, making it vastly more efficient and sensitive. So NAD+ provides the fuel, and resveratrol stomps on the gas pedal.
Got it. So SIRT1 is now fully fueled and highly active. What exactly does it do to the architect, PGC1 alpha, to start building mitochondria?
It performs a chemical modification called deacetylation.
Okay.
PGC1 alpha has an acetyl group attached to it that keeps it locked down and inactive. SIRT1 comes over and literally snips that acetyl group off. That action acts like a key turning in a lock. Once deacetylated, PGC1 alpha wakes up, immediately travels across the nuclear membrane, enters the cell's nucleus, binds to respiratory factors, and begins transcribing the genetic code to build brand new mitochondria.
That is an incredibly elegant chain of command. We fuel the foreman, who unlocks the architect, who goes to the vault to get the blueprint.
Exactly.
But I have to push back here. This all sounds great on paper. But what if you were taking all of the NMN and resveratrol in the world, you were sending the architect to the nucleus every day? But the construction crew shows up to the empty lot, and they don't have any concrete or steel. What if the body is fundamentally depleted of the raw materials needed to build a power plant?
That is the single most common clinical pitfall in longevity medicine.
Really?
Yes. People spend thousands of dollars on NAD+ boosters trying to force biogenesis, but their foundational micronutrients are completely depleted. You cannot build a thermodynamic engine out of thin air.
Give me examples. What basic materials are we usually missing?
Now, let's start with magnesium. Remember ATP synthase, the spinning turbine that makes the energy?
Yeah.
That entire catalytic reaction is magnesium dependent. If you don't have adequate intracellular magnesium, the turbine will not spin, no matter how many new mitochondria you build. And yet, massive portions of the population are clinically deficient in magnesium due to depleted soils.
Wow. What else?
Vitamin D. Over 70% of adults have suboptimal levels. The mitochondrial inner membrane is studded with vitamin D receptors that regulate respiration. You also need a full spectrum of B vitamins, specifically riboflavin and niacin, which act as the core structural components for the electron carriers themselves.
And there is one structural component that stood out in the sources above everything else: coenzyme Q10. The clinics emphasize this heavily, especially because a very common medication actively destroys it.
Yes, statin-induced mitochondrial dysfunction. Millions of people are on statins to lower their cholesterol. Statins work by blocking an enzyme in the liver called HMG-CoA reductase.
Right.
The problem is that exact same biological pathway is responsible for synthesizing CoQ10. By taking a statin, you can inadvertently deplete your body's CoQ10 levels by up to 40%.
Why is that a disaster for the power plant? What exactly is CoQ10 doing inside the mitochondrion?
In the electron transport chain, CoQ10 is the physical bridge between the stations. It sits deep inside the lipid membrane. Its job is to physically accept the high-energy electrons from complex 1 and complex 2 and ferry them over to complex 3.
So if you trigger biogenesis and you build millions of brand new mitochondria, but you are severely deficient in CoQ10, it's like building a state-of-the-art nuclear plant, but you forgot to install the high-voltage transmission lines. The reactors are running, but the power literally cannot get out of the building.
That is exactly what happens. The electrons get backed up at complex 1, the chain stops, ATP production plummets, and worse, those backed up electrons start spilling out into the matrix, generating massive amounts of ROS. You tried to heal the cell, but because you lack the CoQ10 bridge, you just created more oxidative stress.
So supplementation is required.
Yeah.
But not all CoQ10 is the same.
Correct. As we age, our ability to convert standard CoQ10, ubiquinone, into its active antioxidant form, ubiquinol, declines sharply. Taking the reduced form, ubiquinol, ensures the transmission lines are immediately ready for use, and the clinical outcomes are undeniable.
Tell me about the ME/CFS trial from Spain.
A rigorous clinical trial took a cohort of ME/CFS patients, people with profound energy crises, and administered CoQ10 along with NADH. Over 12 weeks, the treatment group showed a statistically significant reduction in both cognitive fatigue and overall physical fatigue scores.
And how do we know that wasn't just a placebo effect?
Yeah.
That they just felt better because they were taking a pill?
Because the researchers didn't just rely on surveys, they actually drew blood. The biochemical analysis showed that the supplementation objectively increased their intracellular ATP levels while simultaneously decreasing markers of oxidative damage. They were quite literally generating more power and producing less pollution.
Okay, so using mitophagy agents like Urolithin A and building materials like CoQ10 and NAD plus boosters is incredible. It feels empowering. But what about the absolute frontier of medicine?
The frontier is where it gets crazy.
Yeah. What happens when a patient's mitochondria are so structurally shattered by a virus or so genetically degraded that supplements just aren't enough? I was looking at the section on advanced frontiers in the notes, and it frankly blew my mind. Let's start with structural stabilization. What is elamipretide?
Elamipretide, commonly known as SS31, is at the bleeding edge of mitochondrial medicine. To understand it, we need to go back to the physical architecture of the inner membrane, the cristae. The molecule that holds those folds in their tight, curved shape is a highly specialized cone-shaped lipid called cardiolipin.
So cardiolipin is the scaffolding holding the walls up.
Exactly. When a cell undergoes severe stress from ischemia, a virus, or heart failure, the immense ROS production oxidizes the cardiolipin. When it oxidizes, the scaffolding snaps. The tight folds of the cristae flatten out and collapse on themselves.
Like the floors of a skyscraper pancaking down during an earthquake.
Yes, and when the floors pancake, the electron transport chain complexes, which were perfectly aligned on those walls, fall apart. Respiration halts entirely.
So how does SS31 fix a pancaked building?
SS31 is a highly unique water-soluble synthetic tetrapeptide. It is incredibly small. When injected, it selectively zeroes in on the inner mitochondrial membrane and binds directly to the oxidized cardiolipin. It acts as an artificial structural stabilizer.
Wait, you're saying it acts like microscopic scaffolding jacks. You inject it, and it physically props the collapsed floors back up.
It does. In studies of freshly explanted, failing human hearts, applying elamipretide actually restored the physical architecture of the cristae. It reorganized the respiratory chain complexes back into their proper supercomplexes and significantly restored mitochondrial oxygen flux and ATP production.
Unbelievable.
It is essentially structural engineering at the molecular level.
Propping up the architecture of a failing heart with a peptide. What about the brain? You mentioned the clinical use of ketones. I think most people hear ketones and they think of the keto diet, weight loss, putting butter in their coffee. Yeah. How does this relate to advanced mitochondrial repair?
The diet industry has entirely obscured the true medical power of ketones, specifically D-beta-hydroxybutyrate, or DBHB. Yes, the liver produces DBHB from fat when glucose is scarce. And yes, it is a highly efficient alternative fuel for the brain, but its most profound role is not as fuel. It is as an epigenetic signaling molecule.
A signaling molecule. What message is it sending, and to whom?
It drives a process called mitohormesis.
Hormesis.
Right. Hormesis is the concept that a small amount of stress creates a beneficial protective adaptation. When you are in deep nutritional ketosis or taking exogenous DBHB, the ketone enters the neurons and acts directly on a sirtuin called SIRT-T2.
Okay, we talked about SIRT-T1 earlier. It's the foreman for building mitochondria. What is SIRT-T2?
SIRT-2 specifically drives transcription factors that promote lysosomal biogenesis. It tells the brain cells to rapidly build massive amounts of cellular recycling centers to clear out plaque and misfolded proteins. And what makes this so revolutionary is that it does this completely independent of the mTOR pathway.
Okay, let's pause. What is the mTOR pathway? I see this everywhere in longevity literature, but no one ever explains it simply.
mTOR stands for mechanistic target of rapamycin. Think of it as the body's ultimate growth mode switch. When you eat protein or lift weights, mTOR turns on. It tells the cell, times are good, we have nutrients, let's build new tissue and multiply.
Sounds great. Why is that a problem?
Because when a cell is in growth mode, mTOR, it completely shuts down repair mode, which is autophagy and mitophagy. The body cannot build a house and renovate it at the same time. This is why fasting is so popular. It suppresses mTOR to allow the body to clean up trash.
So DBHB allows the brain to trigger that deep cellular cleanup without having to starve yourself to suppress mTOR.
Exactly. It bypasses the normal restrictions and powerfully protects the brain against bioenergetic failure. This is why specialized ketogenic interventions are being heavily researched in clinical trials for traumatic brain injuries, Alzheimer's, Parkinson's, and refractory epilepsy. It fundamentally rewires the brain's mitochondrial resilience.
Okay, SS31 rebuilding scaffolding, ketones triggering brain cleanup, but there was one more frontier in the notes that stopped me in my tracks.
The transplants.
Yes, mitochondrial transplantation, MTX. I read this and I genuinely thought it was a typo. Are we literally doing organ transplants with microscopic organelles?
We are. It is arguably the most radical, fiercely debated frontier in modern medicine right now. Doctors are harvesting healthy, viable mitochondria from one tissue and delivering them directly into a host's damaged, failing cells.
Okay, I have so many questions, but let's start with the practical application. Walk me through the pediatric ECMO cases mentioned in the sources. How did they actually do this?
This was pioneered by a team in Boston. They were dealing with infants who had suffered severe ischemia, a catastrophic lack of blood flow and oxygen to the heart during complex surgeries. Their cardiac mitochondria were essentially dead. The infants' hearts could not pump.
Terrifying.
It is. They were placed on ECMO, which is maximum life support that pumps the blood for you. Historically, many of these infants do not survive or they require full heart transplants.
So what did the doctors do?
They took a small, stamp-sized biopsy from the baby's own rectus abdominis muscle, their abdominal wall. They rapidly took that tissue to a specialized centrifuge, mashed it up, and used filtration to isolate billions of perfectly healthy, pristine mitochondria. Then, they loaded those free-floating organelles into a syringe and injected them directly into the dying ischemic heart muscle of the infant.
Just injected raw mitochondria into the heart wall. And what happened?
The clinical results of the pilot study shocked the medical community. 8 out of 10 infants regained enough myocardial function to successfully come off ECMO, compared to a much lower historical control rate. And the recovery time dropped from an average of 9 days on life support to just 2 days.
That's a miracle.
The failing heart cells absorbed the fresh power plants and started beating again.
Okay, the results are undeniably incredible. But I have to stop you here, because mechanically this sounds impossible. I am a skeptic.
Fair enough.
You were just throwing raw engines into the bloodstream. It sounds like tossing a car engine onto a busy highway and expecting a passing chassis to just swallow it up, bolt it in, and start driving. How do these free-floating organelles survive? The blood is filled with calcium, and high calcium usually triggers mitochondria to swell and die instantly.
Your skepticism is completely warranted, and you have zeroed in on the exact controversy tearing the field apart right now. Skeptics point out those exact hurdles. How do they survive the extracellular calcium? How do they not trigger a massive immune response, given they have bacterial DNA? And most importantly, how do they physically cross the lipid membrane of the heart cell to get inside?
Right. A cell doesn't just leave its doors open. How do the engines get in?
The prevailing theory is a process called macropinocytosis. Essentially, the dying heart cell is so desperate that it reaches out and engulfs the mitochondria in a vesicle, pulling it inside. But the exact physics of this endocytosis are the subject of intense, hostile debate.
I bet.
Scientists are actively trying to optimize the delivery to prove it works consistently. They're using MACS, magnetic cell sorting, to isolate purer yields of mitochondria. Some labs are even trying to package the mitochondria inside liposomes, like tiny protective fat bubbles, to help them merge safely through the cell membrane without alerting the immune system.
It is absolutely wild that it works clinically to save infants' lives while scientists are still in a room arguing about the physics of why it works.
Medicine is funny like that sometimes.
But this leads me to my final pivot. As fascinating as mitochondrial transplants are, nobody listening to this is going to get a rectus abdominis biopsy tomorrow to fix their 3 p.m. brain fog.
No, probably not.
If I can't get an experimental peptide infusion, what are the everyday free levers that I can pull to actually change my cellular biology? Because the integrative clinics were very firm on this.
They are emphatically clear, and it is a hard truth for many biohackers to swallow. You cannot out-supplement a bad lifestyle, period.
Right.
You can take thousands of dollars of Urolithin A and CoQ10, but if you ignore the biological foundations, you are pouring water into a bucket with a massive hole in it. The 3 major lifestyle levers that physically alter the mitochondrial grid are training, temperature, and time.
Okay, let's start with training. Exercise. Obviously, exercise is good. But the notes highlight a massive debate in endurance and longevity circles between HIIT, high-intensity interval training, and Zone 2, which is moderate steady-state cardio. At a cellular level, which one actually builds better mitochondria?
It isn't about which is better, it's about the fact that they trigger completely different molecular pathways to build different types of mitochondrial adaptations.
Break that down for me. What happens when I do a HIIT workout? Say I'm doing all-out wind sprints on a track.
When you sprint, you demand ATP faster than the mitochondria can supply it. The cell violently shifts into glycolysis, depleting ATP and flooding the cell with lactate and AMP. This massive energetic crisis triggers a high-amplitude spike in a cellular energy sensor called AMPK.
AMPK, another acronym. What is it?
Think of AMPK as the cell's emergency fuel gauge. When the gauge hits empty rapidly, AMPK screams at the cell to adapt. The specific adaptation to HIR is that it tells the cell to build larger mitochondria, and it drastically increases the activity of the oxidative enzymes inside them. It is optimizing the engines for maximum horsepower and maximum capacity.
Okay, so HIT builds bigger, more powerful individual engines. What about Zone 2? If I'm just doing an easy conversational jog or riding a stationary bike while watching Netflix, I'm not running out of ATP, so what is the trigger?
Zone 2 relies on continuous aerobic fat oxidation. You aren't triggering the emergency AMPK alarm. Instead, the continuous low-level muscle contraction creates low-amplitude, sustained calcium oscillations inside the muscle fiber. This constant drip of calcium activates a completely different signaling pathway via an enzyme called CAM-KII.
And what adaptation does CAM-KII build?
CAM-KII doesn't necessarily build bigger engines. It builds more of them. It radically increases your mitochondrial density. Furthermore, it triggers angiogenesis, which is the building of vast capillary networks around the muscle fibers, to deliver more oxygen to these new mitochondria.
This reminds me perfectly of the high school track runner analogy from one of the coaching sources. If you want a fast mile time, you can't just do intervals every day.
No, you'll burn out.
Right. The easy Zone 2 miles are like compound interest. You do them consistently day after day for months, and they slowly build a massive, dense aerobic base, a massive grid. The HIIT intervals, on the other hand, are just sharpening the edge of the blade. You need both, but 80% of the volume has to be the easy building of the grid.
Precisely. If you only do HID, you have a few massive engines, but you lack the capillary infrastructure to support them long term. You need the Zone 2 foundation.
Okay, the second lever, temperature. Thermal stress is arguably the biggest trend on the internet right now, particularly cold water immersion or ice baths. People are plunging into freezing water every morning. What does that shock actually do to the power plants?
It activates a highly specialized tissue called brown adipose tissue, or BAT. Most fat in adult humans is white fat, which just stores energy. Brown fat, located mostly around the collarbones and spine, is brown because it is incredibly dense with mitochondria.
So brown fat is just a massive heater.
Essentially, yes, but the mitochondria in brown fat do something unique. They contain a specialized protein called UCP1, or uncoupling protein 1. Remember the spinning turbine ATP synthase?
Right, the protons flow through the turbine to make electricity.
Well, UCP1 provides an alternate backdoor for the protons to flow through. It literally uncouples the electron transport chain from ATP synthesis. Instead of the energy being used to make ATP, the rushing protons just generate pure radiant heat to keep your core organs from freezing.
So the body intentionally short-circuits its own power plant just to make a fire. That is incredible. And doing this improves insulin sensitivity.
Drastically. Because to fuel that massive fire, the brown fat violently pulls glucose and lipids out of the bloodstream. It acts like a metabolic sink, clearing out excess blood sugar.
But there is a massive catch, isn't there? Because the sources were very specific about timing. You can't just jump in an ice bath whenever you want if your goal is building muscle.
Timing is everything. We talked earlier about MTOR, the cell's growth mode switch. Clinical studies show that if you do a heavy resistance training workout, you turn on MTOR to rebuild the muscle. If you jump into an ice bath immediately after that workout, the profound cold stress suppresses the MTOR pathway for up to 48 hours.
So you basically freeze the construction crew.
You blunt the inflammation that is necessary for the repair signal. You essentially cancel out a massive portion of the muscle growth and protein synthesis you just worked so hard for. If your goal is hypertrophy, keep the ice bath far away from the lifting session.
Good to know. What about the opposite? Heat stress, saunas. Do they suppress MTOR?
Heat stress acts differently. It activates heat shock proteins. Think of these as cellular chaperones. When the cell gets dangerously hot, proteins start to unfold and denature. Heat shock proteins bind to them, stabilize their structure, and preserve mitochondrial function under stress. Heat is generally highly protective, improves cardiovascular function, and enhances recovery without blunting the hypertrophy response.
Okay, the final lever, and probably the one people ignore the most, time. We are talking about sleep and circadian rhythms. I know I need sleep to feel rested, but do my mitochondria actually care what time it is? Do they have a clock?
They profoundly care. Your mitochondria literally maintain their own intrinsic biological clocks, completely distinct from the central pacemaker in your brain, the suprachiasmatic nucleus.
Wait, the engines know what time it is. Why do they need to know the time?
Because they have to schedule maintenance. The mitochondria conduct critical membrane recycling, repair, and that PINK1 Parkin mitophagy cleanup we discussed, primarily during periods of sleep and fasting. They divide their day into processing fuel and taking out the trash.
So if I am sitting on the couch at midnight, staring at the blue light from my phone and eating a massive bowl of cereal,
you are creating total biological chaos. The light from your phone is telling your brain clock that it is noon. But you are dumping glucose into your stomach, which signals the peripheral clocks in your liver and muscles to process fuel. You are forcing your mitochondria to fire up the turbines at the exact moment their local clocks are trying to shut down for scheduled maintenance.
I am forcing the factory to run at maximum capacity while the janitors are trying to mop the floors around the machinery.
Exactly. It drives massive metabolic confusion, insulin resistance, and profound oxidative stress. Furthermore, there is a paradigm-shifting discovery regarding melatonin that ties this all together.
Right. I wanted to bring this up. We talk about melatonin in the ancient bacteria. Everyone knows melatonin as the sleep hormone that the pineal gland makes in the dark.
That is the endocrine function of melatonin. But recently, researchers discovered that mitochondria actually synthesize their own local melatonin right inside the mitochondrial matrix, totally independent of the pineal gland.
Why are they making a sleep hormone inside the engine room?
They don't use it for sleep. Melatonin is arguably the most potent ancient antioxidant in biological history. The mitochondria synthesize it locally to act as a highly concentrated, localized shield against the ROS leaking from the electron transport chain. But this localized synthesis is tightly linked to your circadian alignment and your exposure to natural light during the day.
So if I have poor sleep hygiene, if I am staring at screens late at night, if I am chronically stressed and pumping cortisol, I am actively disrupting the production of the very shield that protects my power plants from rusting. I am putting a hard biological ceiling on my cellular energy capacity. I can take all the Urolithin A, I can take the NAD+, and the CoQ10, but if I don't respect the clock, I am constantly tearing down the grid from the inside.
That is the fundamental truth of metabolic rehabilitation. Lifestyle provides the foundation. It sets the clock, it builds the base density, it creates the environment. The supplements provide targeted scaffolding and cleanup once that foundation is laid.
Man, what an unbelievable journey this has been. Let's recap this, because it completely changes how we have to look at our daily fatigue. We started billions of years ago with an ancient endosymbiotic merger, a cosmic accident, that created the localized cellular power plants we rely on for every heartbeat and every thought.
We explored how processing oxygen, while efficient, is a thermodynamically messy business, leaking ROS and causing a slow accumulation of oxidative stress that literally defines the aging process.
We looked at the dark side of this machinery, how chronic infections like the SARS-CoV-2 virus can physically shatter the cristae scaffolding, throwing patients into a profound acquired energy crisis. We define PIM not as tiredness, but as a catastrophic failure to scale energy, leading to toxic glycolysis and systemic inflammation triggered by leaked, ancient bacterial DNA.
We saw how the body prioritizes survival above all else, with starving peripheral muscles stealing glucose from the brain, plunging older adults into cognitive decline, and how restoring glutathione with GlyNAC can halt that bioenergetic robbery.
We talked about calling in the autophagosome garbage trucks with Urolithin A to clear out the condemned buildings, and then using NAD plus precursors, resveratrol and CoQ10 to wake up the architect, supply the materials, and build brand new, highly efficient mitochondria.
And we've ventured onto the absolute frontiers of medical science, looking at cardiolipin stabilizers, propping up collapsed architecture, ketogenic signaling molecules triggering brain cleanup, and the mind-bending reality of injecting free-floating organelles directly into a failing heart to save a child's life.
All anchored by the reality that we control the daily levers. Zone 2, training to build the grid, thermal stress to tune it, and respecting our circadian clocks so the engines can finally rest and repair. It is a completely radical way of viewing human health.
It is, and I want to leave you with a thought that really underscores just how profound this perspective change needs to be. We just spent an hour discussing how mitochondria have their own DNA, completely separate from ours. They maintain their own localized biological clocks. They synthesize their own ancient hormones like melatonin. And through the process of apoptosis, they are the entities that ultimately pull the plug and decide when a human cell lives or dies.
That is a staggering thought to end on. It really makes you wonder, are we, the consciousness listening to this right now, truly in charge of our bodies? Or are we just highly evolved, walking meat suits built over billions of years for the sole purpose of serving and transporting trillions of ancient endosymbiotic bacteria? Think about that the next time you pour a cup of coffee. Thank you for joining us on this deep dive into the cellular power grid.